Glad you joined us for our webinar today with Sarah. Again, she'll be presenting tips for performance evaluation planning to support your transition from IVDD to IVDR. All right, our presenter today, as I said, is Dr. Sarah Chavez. Dr. Chavez is the Director of IVD and Scientific Writing Services with Criterion Edge. Sarah has a BS in biochemistry and a PhD in molecular and cellular biology with 25 years of laboratory bench experience and has taught at the university level for nearly 20 years. She has extensive scientific writing experience in an academic environment as well as for several large textbook publishers. Sarah's strong background in basic research and her extensive experience with regulatory writing will allow her to assist companies with IVD readiness as part of the Criterion Edge team. Now I'm going to hand this to Sarah, who will be presenting our topic today. Sarah?
Okay, thank you, Mark. So the topic for today is this whole concept of performance evaluation. And we realized early on in helping companies with this transition that this is not a new concept, but the actual requirements under the IVDR are going to catch some people off guard. In particular, there are these two new documents, the Performance Evaluation Plan and the Performance Evaluation Report, that seem very daunting for someone new to this field. So in order to help manufacturers better prepare and be in compliance with the IVDR, we wanted to pull together some of the tips that we've learned through the early implementers for all of you to employ as you embark on your own IVDR submissions. So today we're going to first just provide some definitions in terms of the scope and objectives of performance evaluation planning, and discuss a little bit about systematic literature review, which is one key component of the PEP that seems to be really catching people off guard. We then will discuss things like the scientific validity report, the analytical performance report, and the clinical performance report, and discuss basically some strategies that you can use to help you compile these data and pull them together into a cohesive package within the PER. So let's start with just a few basic definitions, just to help make sure that everyone is on the same page. Performance evaluation, ironically, it's not actually defined in the directive, but it is well defined in the European Harmonized Standard, which states that it's the performance evaluation itself is an investigation of the performance of any in vitro medical device based on data already available, scientific literature, and or performance evaluation studies. So a lot of you in the IVD world are very concerned about this last one, those performance evaluation studies. So hopefully by the end of today, we'll be able to walk you through some potential ways of utilizing that data already available in the scientific literature. When they speak about performance evaluation studies, what they're really talking about are these new investigations of IVDs intended to validate the performance claims under the anticipated conditions of use. And that's where aspects like the population, indications, comparators, and outcomes, and ultimately your intended purpose feed into that. So really, the whole performance evaluation process, within these performance evaluation studies represent those investigations that are designed to help you fill in some of those gaps based on your Picoterms. So it's really critical to define your Picoterms very early in this process. So as you're beginning your performance evaluation, the PEP should be constructed to allow the evaluator to correctly perform and appropriately report on the performance evaluation overall. So a proper PEP should cover at least these aspects. So details on the intended purpose or intended use, the performance specifications of the IVD as established by the manufacturer, basically to ensure that that intended purpose is actually being fulfilled. You'll also want to describe the analyte and or whatever biomarker that is being measured by your IVD, and any related reference standards or ways of ensuring traceability for that particular biomarker. You also need to be very clear in the way that you identify your specific target patient groups, along with your indications and any limitations or contraindications. It's also important to take into account things like the global safety and performance requirements that your PE will support, as well as providing details on the methodology, including things like statistical tools that you're going to use to assess the analytical and clinical performance of your IVD. There are also these aspects of this development phase. So you want to provide an overview of the developmental phases of the IVD with milestones, acceptance criteria, and also indicate any sort of timing as far as your validation and verification testing. So ultimately, this is going to contribute to you compiling this state of the art, this benefit risk ratio, and then eventually you're going to get into your PMPF planning. So, just a few basic considerations and things to take into account. Early on in your process, you want to ask yourself how much research is really out there on this device or devices like mine. Is analytical performance already completed? Are there any proof of concept studies? Even if they're preclinical, they can still be relevant to the scientific validity of your report. Um, and then finally, you want to see, is the available data that's out there as I'm beginning to search for it, is it the right level of good laboratory practice? And do I think that the results of my analysis in the published literature is going to be sufficiently robust to support my performance evaluation? So ultimately, that's where systematic literature review comes into play. Systematic literature review is nothing new. Anyone in science has done these a dozen times over, even during graduate school. So it's a very common assessment that you use, but it can be overwhelming given that this is a brand new requirement under the IVDR. And the notified body is expecting that you're going to report the results of your systematic literature review in each of your reports and document that process so you can ensure it's reproducible, transparent, and unbiased. So we want to define our questions and be very precise early on. And there are multiple research questions really that are driving our searches. So that's where that clear definition and alignment with your intended purpose statement is going to help you refine your search strings to better delineate between the purpose of the searches that you're going to conduct. Because the results of these searches will contribute to the data sets that are going to feed into the various reports. So, for instance, your SVR, scientific validity report, should aim to justify the acceptability of the analyte and the testing methodology. So, you're trying to prove what is state of the art for whatever your intended purpose is. You also want to define the context for measuring success and any endpoints that you're going to use, any performance measures. That's where the analytical performance comes into play. And remember, you've assigned these limits as to who is your population and the indications and the outcomes. Those terms, those PICO terms, are really going to help you and need to be justified within that clinical performance report. So, really, while that state of the art search is just one search, it should help you focus on the PICO terms, and then you'll develop a secondary search that's going to be more focused on any professional guidelines or recommendations, anything that's going to help you really justify and establish the positioning of your device within the market as a whole. The competitor search, though, that can be a little bit more targeted, and that's where knowing your competitors, including their specific product names, could help you capture relevant published data in support of developing acceptance criteria. The subject device can be a separate search, and that data is usually not incorporated as part of the state of the art, except in the case, in rare cases, where you have a very novel biomarker and you kind of are the only thing out there in that arena. But for the most part, the state of the art section, the scientific validity section, is being presented regardless of your test being there. You're sort of establishing what is the state of the art and where do I fit? Where would I fit within the state of the art? So, this entire process requires a very well documented strategy and methodology. So by the time you get to screening, that can be extremely time consuming, even under the best of circumstances. So I definitely encourage you to take your time with that. We actually recommend this multi-level approach. So you perform the search, and as you're screening the articles, first look at the title and the abstract and see if any of them can be eliminated at that level rather than jumping straight to the full text for every single article. Extracting that data can also be really time consuming, but if you've established the correct and most appropriate analytical and clinical performance objectives, that's going to really help you narrow down and search for those exact data pieces as they're presented in published literature. So it's helping you make sure that you are identifying the most relevant performance objectives. When it comes to appraisals, there are certain accepted methods, including those published by the Global Harmonization Task Force and level of evidence, as we've shown here. But be prepared to justify the articles that you've chosen to include and exclude, and follow best practice for that data collection piece. And then ultimately, you have to report on your process. So this is typically represented by one of these Prisma diagrams, which is a visual representation of your entire process from searching through screening and ultimately presenting the results. But this is going to need you to clearly document the search strategy, exclusions, inclusions, and the appraisals. So remember back to your high school writing classes, before you start writing, you have to have that research question. You have to know your purpose. Why are you writing this paper in the first place? And you also have to know your audience. So in this case, we're lucky in that we know our audience, the notified bodies and the reviewers, but we have to ask ourselves about the purpose behind the searches. And it goes back to that primary objective of performance evaluation. We want to describe and provide an assessment and analysis of data to establish or verify scientific validity. And that establish or verify is tricky because sometimes scientific validity is well established. And other times you may have a novel biomarker where you are actually providing data to help you verify a new or emerging unestablished biomarker. You also have to have analytical data and, where applicable, the clinical performance data of the device. So, in order to accomplish the goal of collecting these data pieces, you need to have a way of finding that clinical evidence. In terms of the IVDR, they refer you back to the intended purpose on multiple occasions in their guidances. It also mentions that performance evaluation is this continuous process. So that's a good way to start thinking about this. The PER is a product. It's basically a snapshot in time of what is the current state of your device. But the process of collecting this data is continuous, and you should be prepared to have a methodology so that if anything is changing, if there is new information emerging, if there are new biomarkers that are perhaps more sensitive or more specific, you want to be on top of that. So that's ultimately why this entire process and this mandate has come about in the first place. So there can, in some cases, be some of these significant changes in design or intended purpose. And if you're ultimately developing your searches, you need to be prepared to make those changes if your intended purpose potentially changes along the way. Clinical and analytical data obtained through new studies and newly published information could ultimately prompt a significant change. So, for instance, if there is a change to the diagnostic sensitivity, or if you identify a new potential cross reactivity or interference, those should be fed into your instructions for use, and that could be considered a significant change. Manufacturers should be monitoring this post market data primarily to ensure that the device is being used for its intended purpose, because some of these off-label usages in the literature could trigger updates to your labeling or new warnings or limitations that could also result in the need for an updated IVDR application. And it should be noted that limiting your intended purpose, perhaps by removing a particular associated clinical condition or a claim, That's not necessarily considered a significant change, but if you wish to go back and expand your intended purpose to a new clinical condition, that could be considered significant. So, really aligning your intended purpose based on these aspects, whether you're intended for screening, diagnosis, classification, prognosis, etc., aligning your intended purpose with the available data sources you have at your disposal will help you define the purpose of the intended searches, but it also helps you with thinking about how you're going to organize all of this information into each of those required reports. So now I'd like to dig into basically what are some of the strategies that you can undertake as you're approaching these standalone reports. There is a guidance in case you haven't gone to this one yet. This guidance is very helpful with providing some of the basic definitions we need. It's this MDCG 2022 2. It provides us with a guideline to follow to help us establish scientific validity. And that's really going to put us on the right track because that ensures that the overall approach taken to establish that association between the analyte and the condition is a valid one based on the state of the art. So I always recommend doing the scientific validity report very early in the process. It is really going to give you a good sense of the types of performance measures that are widely used and any sort of things to be on the lookout for and kind of figuring out that competitive landscape. That's going to be really valuable as you're preparing to write your analytical and performance reports for your device. It's nice to know what else is out there for your competitors or for your subject device for that matter. So, scientific validity can be demonstrated through the use of this existing data whenever it's available. And it should take into account what's generally acknowledged to be state of the art. And that's based on professional recommendations and guidances, but also just the sort of consensus within this peer reviewed published literature. If the association of an analyte to a clinical condition is well established, you may have sufficient quality and quantity of evidence to substantiate the scientific validity in a fairly short manner. It shouldn't take that much to establish some of these biomarkers. In such cases, that association may be more appropriately demonstrated based on that peer-reviewed literature, textbooks, historical data, and professional experiences. If there is no existing evidence, or if existing evidence is what you would deem to be insufficient, there could be, and this could be as a result of having a novel device, like a new analyte or a new intended purpose. That's where it really takes a little bit more time to prove the scientific rationale and to generate new or additional data, including some of that preclinical data. So, conducting a gap analysis early in the process is really helpful to determining whether or not you're going to have sufficient evidence in the published literature. The existing data sets can be anything from appraised literature, peer reviewed data, published clinical data, registries. There's a variety of different places you can go for existing data sets. But as they note in the figure they provide here, this is an iterative process. It's not linear. So the information you're gathering during the course of your review could ultimately result in changes or refinements to your intended purpose or scope or conclusions. So really that's why I'm finding a lot of companies begin with the SVR so that you can have a better sense of any changes or potential changes and their implications for planning your clinical performance studies. So the next step is your analytical performance. And at this stage, you really just have to show that your device does, in fact, do what you claim it does. It can detect an analyte using a technology or an approach that's valid. So, in essence, you want to prove whether or not that device detects the analyte in a controlled laboratory setting. The APR or analytical performance report really addresses three main issues or areas. These assays, and that's going to include things like the type of specimens, the storage or transport, and also any sort of reference method or reference standards that you're going to use, as well as potential for interfering substances or cross reactivities. It's also important to take note of the measuring range, and the expected range in your given population, because that could vary for different populations, and that should be noted. There are, in our experience, there are a lot of these aspects that the companies are already prepared for. This is pretty much what IVD companies have been doing from day one. So they're really confident with navigating these aspects. These are kind of the fundamental experiments and analyses that are used to establish that your assay is performing as intended in these laboratory settings. So you will likely have many of these proof of principle studies that have been conducted in-house at the time that the assay was validated. And now conversely, when it comes to clinical performance, this is the tricky part because clinical performance is really that real world data. And the purpose behind a clinical performance study is to establish or confirm the aspects of the IVDs that you can't accurately determine by analytical performance studies alone, or even by the literature that's been published to date. Sometimes if you have a well established device, there's a long publication history, and you will have a lot of published literature and real world data to pull from. We don't all get that lucky. The clinical performance is really that relationship between the testing results and sort of the unpredictable aspects of your clinical conditions for your patients. So you want to take into account as you're looking at clinical performance studies whether or not they have sufficiently accounted for potential risks in terms of incorrect information, false positives, and false negatives for the patients, and also the appropriate ethical principles if you're designing your experiment from the beginning. So really, clinical performance studies should be designed, conducted, analyzed, and evaluated so that you can really get the best possible representation of the target population in line with your intended use. But according to a lot of the early feedback we're hearing from notified bodies, clinical performance studies are the most typical gap in the technical documentation reviews. And that's primarily because clinical performance studies up until now were performed to meet the requirements of IVDD, not IVDR. So the majority of existing or legacy devices rely heavily on scientific peer-reviewed literature as the main source for their clinical performance data. But if you have limited published data, that could put you at a severe disadvantage. So, where do you even begin to start for looking for clinical performance data? These are where you're going to find your in house clinical performance studies, the peer reviewed literature, and then published routine diagnostic testing. That can be very time consuming in terms of searching and screening the literature for that published routine testing. Because ultimately, in many cases, your device is not the subject of that article. The purpose of the article may be unrelated to the testing or sort of tangential to the testing itself. So you may have to review the full text to really find examples of your device being used in real world settings and then having an impact on patient outcome. Do the results of your test really support the diagnosis or support some aspect that's going to have a direct clinical impact? So that's really why clinical performance is considered distinct from scientific validity. It must be valid for European practice too. So you wouldn't want to use any outdated standards in terms of the guidances that are published in the EU. So paying attention to the up-to-date recommendations and requirements, that's going to be really critical. The PE plan should reflect clinical performance today based on the current intended purpose. So it's not a history of every single thing that's ever been used with your device. You want to look at the state of the art. You want to look at the most current reflection of your device and its intended purpose. So you want to be able to justify the strategy you're taking in terms of your performance evaluation reporting. So on the next slide, I have tried to pull together some tips and tricks for choosing performance objectives wisely. Um, In a way, the IVD companies actually have it a little bit easier than the medical device companies because we are fairly uniform in terms of how we can measure success. Analytical performance in particular, these are going to be things like precision, reproducibility, accuracy. These are all aspects of that sort of quality testing, making sure that a test works as you intend it to under ideal laboratory settings. The analytical performance studies focus on the analyte, and the clinical performance studies are focusing on the patient perspective. So they are inherently related, but because really that analytical performance is the basis of the clinical performance. If it's not working clinically, there's probably a basic science reason for that. But the analytical performance data don't demonstrate clinical performance. They tell you it works under ideal circumstances that you've anticipated in a laboratory. But once it gets out into the real world, does it still work? Your test may perform with accuracy and reproducibility under those control settings. But are there situations or particular patient groups in which the test may not perform as well? It's entirely likely. What if your patient is already receiving a particular treatment? Could that affect the levels of analytes that are detectable for that patient? So therefore affecting your any potential for identifying lower ranges, expected ranges. Are there characteristics of certain types of tissue or samples or some other reason why the test may not perform as well? You want to identify whether or not a patient is at an increased risk of getting a false result, either false positive or false negative. So I wanted to just sort of insert a really quick example just to provide you kind of a real world example of how this could happen. So in 2013, the American Society of Clinical Oncology and the College of American Pathologists had recommendations that stated that if you are making a diagnosis using FISH for HER2 positivity in breast cancer, you should include the average HER2 gene number per tumor cell, as well as a ratio of the average HER2 to an internal control gene. And they gave, in this case, it's chromosome 17 centromere was the internal control. And based on sort of their recommendations, there were 5 possible outcomes for or clinical scenarios. One of those groups was described as something called HER2 equivocal, meaning that when you got the results back, you couldn't tell there was neither an amplification of HER2 or a decrease. It was just sort of equivocal. It didn't appear that there was any change in HER2 levels. By the time we got to 2018, there was a clinical practice update that cautioned that this indiscriminate use of these alternative internal controls could lead to a significant number of positives. And so they actually recommended including IHC as a way to resolve those cases. So now rather than saying you can stop and say it's equivocal, now they're saying, no, no, let's do a further confirmatory test. And actually, when they assessed this particular strategy, they realized that when you used recommended alternative control genome sites in many patients, there were quite a few of patients who were inappropriately diagnosed as being HER2 positive, which could impact the type of treatment you're receiving. Those tests performed precisely as intended in an analytical sense. So everything was fine in the laboratory. The tests were working as expected. But once you got into the real world clinical setting, some of those controls that they had identified in the testing phase resulted in substantially higher rates of false positivity and impacted the clinical care for those patients. So, real world data does matter. And that's why we have separate batches of data that we're reporting on for both the analytical as well as the clinical. Now, this is the other question that a lot of groups are really concerned about. It's that data sufficiency. Everyone wants to know how much is enough. When you think about data sufficiency, consider not only quantity, but quality. The quantity is that's typically going to be dependent upon whether or not that test is established or standardized. Your device may be widely considered as, you may even be a benchmark. You may be an accepted option. Or ideally, it could be specifically recommended in professional guidelines. That's really going to help you build your argument for being considered state of the art. But sometimes your tests are not necessarily so well accepted, and you really don't expect to find a ton of published data over the most recent 5 years. But that's okay. So the Global Harmonization Task Force anticipates this, and they help you have a better understanding of what types of studies are going to be most appropriate based on where your device falls. If you are established and standardized, those are the assays whose analytical performance combined with scientific validity meet the clinical evidence requirements. And so that's sufficient to demonstrate compliance with the essential safety and performance principles. Those tests have associated clinical guidelines or consensus for their use and produce comparable results for the analyte, regardless of which manufacturer's test you use. So those could be things like liver testing, like ALT or bilirubin tests, or a creatine test for kidney functional markers, electrolyte tests, those sorts of things. But you could also be established but not standardized. And those not non-standardized tests have information about analytical performance, but they don't necessarily demonstrate that same conformity with the essential safety and performance principles. So they could include clinical guidelines or consensus statements, but if you use tests from different IVD manufacturers, they may not be able to be used interchangeably because that standardization has not occurred yet. So, in many cases, if you have an infectious disease assay, you fall under this established and non standardized category. But even things like hormones or cardiac markers, tumor markers, those sorts of things can also fall under this category of established but not standardized. And the third group, that's going to be even the hardest as far as sufficient amount and quality. Those are the novel tests where you are still proving that analytical performance in conjunction with the scientific validity. So you are going to have to spend a little bit more time describing the analyte, the new technology that you're proposing, or the new intended use. But you're neither established nor standardized. So this could be like a newly identified cardiac biomarker or a newly identified tumor biomarker. Or SARS-CoV-2 would also fall under this category. In some cases, you may be lucky and you may have found a systematic review or a meta-analysis sort of laying the groundwork for how you identified that biomarker in the first place. Guidelines can sometimes also include some summaries of the systematic reviews that they used in support of establishing the guidelines. So you can use those to support your own analyses. And those are also considered to be a high quality of evidence and will typically report on any data limitations or potential for bias. But really, it comes down to that balance, identifying whether or not your device falls into a category where you should anticipate that the quality or the quantity of the data could impact your timeline. And then finally, this is where it gets really tricky is that you want to connect between these reports. They're asking you to write standalone documents, but there should be a carry through. You should be able to make connections so that it's clear how your argument fits together. The PER is not the equivalent of just stapling the reports together and sending it on its way. It would be like saying, your doctoral thesis is just stapling all the papers you've published together and hoping that they can read between the lines and see the connections and the story you're telling. They don't do that. They want you to connect the dots. So, in our experience, there are a few common gaps that we see in creating this thread and this narrative, and they really tie back to that state of the art section. And this is especially the case for companies that are undergoing that transition between IVDD and IVDR. So it's really important early on to think about things like your methodology for your searches and how you're going to be choosing your PICO terms. So identifying the correct population indication comparators and outcomes that can be supported by the data. and can be considered to be state of the art based on the current state of the research, not the research 20 years ago. You want to know what's the current state of the art. When you're identifying those PICO terms early, those terms help you guide your literature searches and inform on your choice of alternatives and competitors. So the competitive landscape is going to be really important because that's where the data for the performance indicators or objectives for your competitors is presented. And that's the data you're using to define your acceptance criteria. So your APR and your CPR will present the data with regards to your device, but the state of the art essentially presents options for if your test were not available. And the range of what is considered to be acceptable performance based on those alternatives and competition. So it's really important to ensure that the performance indicators that you're reporting for your competitors align with the guidelines and the professional standards, but also that they align with the performance indicators that you are collecting as part of your own post-market surveillance or internal pre-planned clinical studies. So remember, these documents here, they represent standalone reports developed during the performance evaluation process. They're each supported by independent searches with unique purposes. But the PER, the reporting, should connect those data and ensure consistency between the reports. So be prepared to align the style and the design decisions between the reports to make it easy for the notified body to follow along and to see your argument. A lack of consistent data supporting those scientific validity, that early state of the art section, is going to get noticed very quickly. And it's really hard to disguise that or justify it when the notified body notices that. So I just wanted to conclude with a statement that was taken directly from MedDev 2.7 Rev 4. And this is in reference to a clinical evaluation report, sort of what happens on the medical device side. It says the clinical evaluation report should contain sufficient information to be read and understood by an independent party, such as a regulatory authority or notified body. So sufficient information to be read and understood. Don't make them make any connections themselves. They want you to lay out the argument for them very clearly and even state the obvious so that they don't have to extrapolate or make inferences. So as you're evaluating the own state of your performance evaluation, ask yourself before you get too far along the way, is there going to be sufficient information? And then as I'm writing my report, could someone who is an outsider, an independent third party who is not an expert on my device, could that report be read and understood by that individual? They are not going to know your company's devices as well as you do, but they should feel like they have a solid understanding of where your device fits into the grand scheme of things. So really it comes down to sufficiency, but also understandability. Like can they follow along with the argument you're presenting for your performance evaluation? Okay, and I think we have some time left over at the end for some questions. Really, our goal is to help folks understand really the impact of these IVDR regulations that's going to help you navigate those complexities of compliance and really make informed decisions early on during your development process. I'm hoping this provides some better context as we're moving into the Q&A portion. And I know we had some Q&A questions coming in during the registration process. And so we've pulled from some of those that we've gotten to
Yeah,
from
thanks,
people.
Sarah. I'll
Yeah.
now take us through the questions. As you said, that we have some questions that submitted during registration, but we want to make sure the audience feels free to enter questions as much as possible. So please submit those using that Q&A button that you guys should all see in the Zoom controls. And of course, if you have any issues finding this or anything else in the webinar, please chat and let us know and we'll help you. So with that, I'm going to go ahead into our first question from registration, Sarah. We have that here. What is the most common question notified bodies are asking regarding the literature review process in your experience?
So early on, kind of the trends that we're seeing is really how you've designed your search. Like, how do you identify the competitors that you're choosing to compare yourself against? And how do you ensure that your search is robust enough to capture some of those off-label uses, which they want to make sure you're looking for? Um, but they also want to make sure that you're not searching for everything. Like if you are looking for HER2, you're not just going to search for breast cancer markers. You're going to get 20,000 hits easily. So instead, what they want you to do is design these searches in a very strategic way so that you can follow the train of thought as to what your original research question was, but also understand what are professional organizations and what sort of the scientific community, what are the general consensus surrounding your device. So I would say it comes down to questioning how the searches are conducted, and the choices that are made in terms of developing the search strings. And then also the reporting. A lot of times people are not being super transparent in reporting the methodology. They want to see the reasons for exclusion for every single article. If you have excluded an article, you should be prepared to justify why. If you've included an article, you should be prepared to either cite it or appraise it if it's contributing to your acceptance criteria. So it's about being really organized with this so that they can follow your train of thought and they can see, oh, that search makes sense to me logically. I can see why they designed it in that way.
Excellent. We're going to go on to question number 2 from registration, but again, we're starting to get questions from the audience, so we'll proceed with those next. Second question is, how can you conduct an effective literature search for low-risk devices with limited clinical data and literature?
So if if there's going to be limited clinical data, this comes up for a lot of sort of the tried and true, like the tests that we've had on the market for pretty much forever. You're not going to see people publishing on the clinical performance of pregnancy tests on a daily basis. We assume they work. What you can do though is look for, you know, uh, i- potential ways in which the device may be used in a real world clinical setting. So, you would want to look for the names of the device. And I think this ties into one of the questions we have in the chat here too. If you have a legacy product and you're not finding anything on your own device, or if you're not finding anything on your competitors, like what can you do about that? Um, First off, you want to show them that you looked. Like that's important. You might not find anything. You may conduct a search and the results are zero. As long as you can justify that through to the notified body, that's what they're looking for. But then also take a step further and say, like, are there any other ways that this could be presented within the literature? Maybe I have to do a little bit more digging, but really those guidelines and the professional recommendations, anything that's going to establish sort of the clinical standard, like what really happens in real life. When would you expect this test to be used and under what circumstances? That state of the art is going to be super relevant for those sort of like legacy devices, or they're trying to make it to where you can't just say I've been around forever and so I continue to be state of the art. You really want to show the positioning of your legacy device as it stands now, not when it was developed, but right now. So knowing that there hasn't been a lot of clinical literature published in the recent history is okay. But you may be asked to perform additional clinical studies in the future if there seems like there's a knowledge gap, if you're using like a specific new or expanding the intended purpose for your device. So I think it's important to take a look at your intended purpose statement as a legacy device and just make sure that the published literature for the state of the art truly reflects what you consider to be your intended use.
Right. Thank you, Sarah. We got another question from the audience. Do you have any suggestions about how best to collect and present data on similar competitor devices?
Yes. So when you're ultimately trying to set like sort of an acceptability range. You want to show if my device were not available and the physician was pulling some other test off of the shelf, what would they use and how accurate or precise or how good are those tests? So as you're presenting the data, it kind of comes down to the style of the data and the type of the test. In most cases, if you have multiple ways in which the data is reported in the literature, if it's like a diagnostic indication and you expect to find data on sensitivity, specificity, false positives, false negatives, you wouldn't want to list that in a paragraph form for every single article you came across. No notified body is going to want to read it like that. But presenting it in sort of a tabular format and then summarizing the results can be really helpful. It's just important to recognize that as you're presenting it in a table, if there are any particularities about this is in this tissue or that tissue, if there are any differences between the studies, you would want to capture that in the tabular form. But let's say you have a type of test that is legacy and there's just not a lot of data on anything out there. The narrative may be your best strategy there. You can say, here are some guidelines that regularly recommend this device. Here's, the alternatives. We can clearly see that they don't provide as good of a result. So, forming your competitor narrative or your competitive landscape in more of a narrative form, I find tends to work really well for the legacy devices compared to like or a novel device where there's a little bit more nuance in the data and you need to take a little bit of time to connect the dots and make that explanation. So if you've got a lot of data, put it in a table and summarize it below. If you've got little bit of data, really connect those dots and lay out the argument in a well constructed narrative.
Excellent. So before I go on to the next question, I just want to remind everyone that you could submit any questions you have via the Q&A button in the tools bar. I also want to remind you that you can reach out to us at consult, that's CONSULT at criterionedge.com to schedule some time to talk with any of our specialists. We're available to chat with you about your current or upcoming project needs or answer more confidential questions in a free consultation.
Mhm.
You can also book a meeting with us anytime, the link in the chat. And with that, I'll go on to the next question. Sarah, can you please comment on tips for reviewing preclinical data and determining its relevance for clinical evaluation purposes?
Yes, so the preclinical data, in many cases, if it's a well-established device or technology, we often use non-human or preclinical data as an exclusion criteria. We don't want that data. But if you have a novel biomarker, or if you're trying to describe sort of where your device fits into the broader scheme, if you're still trying to establish that scientific validity, that preclinical data can be really helpful. It can show you what's happening in the preclinical models and sort of extrapolate that, the data is still coming in. We're still learning more in the human testing. But based on the preclinical models, here's how it's going to fill a gap in the available technologies. The available technologies don't fill, there's this unmet clinical need. And based on early preclinical data, this could help us fill that unmet clinical need. So again, it kind of goes back to that state of the art there.
Excellent. We have another question here. Is it always necessary to have clinical studies to support the clinical performance of IVDs?
Oh, yes. So they are going to expect clinical studies if you are not a very low risk device. For some of the class A's, can be exempted from clinical performance studies. For the most part, anything that would be sort of a class B, class C, and especially D, you're expected to have sort of increased requirements for clinical data. Again, that sort of ties in perfectly, good timing, with the question that's in the Q&A, for like a new product that's come out, and you're writing a clinical performance report. They only have a clinical report that's been done in-house, presumably, and there is no published literature. We actually see that quite a bit. If it's a novel product, no one should be publishing on your device if it's not out there yet. So that's to be expected. But what you could do is say, you want to make sure that you've justified in your clinical performance report the study design and the different considerations that went into the design of your clinical study and how that aligns with the current state of the art. So you look at the published literature for your competitors and your alternatives. You look at the types of performance objectives and the data that were captured, and you ensure that the patient population that you're claiming, all of your intended purpose, is fully captured within the clinical study design. So like you would, if your clinical study only includes adult patients, you wouldn't claim a pediatric indication. You're going to want to make sure that you're sort of in a position where your clinical data has to be supported by either published or in-house data. And so if it's in-house, you have both the blessing and the curse of being able to sort of frame the experiment to really drill down on your intended purpose. But sometimes if you are claiming a very broad intended purpose, that could require additional clinical studies to really fully support them.
You, Sarah. Well, we're running a little short on time, so if you have any final questions, please put them in the Q&A. But we'll go to this one we have. How stringent are the notified bodies when reviewing the acceptance criteria for clinical performance?
So yeah, the clinical performance is definitely, I think, what we all were worried about, and rightly so. That seems to be the major gap that a lot of organizations are having, because in many cases, there is no published clinical data. And they really don't want you to say, there's no published clinical data. We haven't done our own studies, or our own studies are 100 years old, but we're still state of the art. They want to see that you have some sort of a bar by which to measure success. So the acceptance criteria in those cases can be largely attributed to sort of those recommendations and guidances, or the published data on the competitor devices that are most closely aligned with your intended purpose. So it comes down to choosing the right competitors to kind of establish the acceptability ranges for performance for your device. But yeah, they're definitely going to be looking for how you've organized that argument and how you've laid out your argument there.
Excellent. Thank you for that, Sarah. It looks like we've come to the end of our time. Thank you, Sarah, and all the participants for the excellent questions. Our team will be sharing some links in the chat and will remain on the webinar for a few more minutes. You can access these. If you've enjoyed our webinar today, you may want to sign up to be notified of additional upcoming webinars produced by Criterion Edge. You can do so by clicking the link in the chat. If you still have questions or on the content today we discussed, or if you'd like to learn more about our services, you can schedule a free appointment with our specialists. As we said before, contact information is consult@criterionedge.com, or you can book a meeting at a convenient time for you with a link in the chat. The webinar is recorded and will be available soon to watch on our website and sent out to you in an email with the slides that we went over today. When you exit the webinar, please take our short survey and let us know how you enjoyed today's talk and what other topics you'd like us to cover in the future. We'd love your feedback on that. We want to make content that's relevant to you. Thank you, everyone, for attending and for all your questions. We hope this presentation gave you more insight for your submissions. Have a great rest of your day, and we'll see you next time. Thank you, Sarah. Thank you, attendees.